Okay, welcome. Uh, Switching it up the littlest bit. I had a have a meeting scheduled for the same time as lecture, so I thought I'd give you a recording. So we left off last time. I know we showed this image talking about the transfer or tRNAs. Just a quick recap with this. It's a couple of different images. We're talking the same thing. Our transfer RNAs in blue look like they're double-stranded, but they're really a single-stranded RNA molecule with it folding back in complementary pairing on itself to make it look double-stranded. The top bit of it has amino acid attachment site. This is going to allow for the different amino acids to come in and bind to this part of the transfer RNA. There's going to be different amino acids linking up onto different tRNAs, and they're going to recognize the messenger RNA, as you see down here in pink. They're going to recognize this messenger RNA based off of the anticodon that is seen on the transfer RNA. And if they match up complementary pairing and hydrogen bond, that amino acid is ultimately going to get donated to the growing polypeptide chain, which we'll see that process happening here in just a second. So we showed the amino acid table and talked about some of the redundancy with that. There are individual transfer RNAs for the respective amino acids, but with that said, we can go back to this and see the leucine. I mentioned a single tRNA is bringing leucine into this mix. The last position of the codon, the wobble with the tRNA means that it doesn't matter as long as there's a C and a U, leucine gets brought in. Up above, there's another way to make leucine, and so a different transfer RNA, because we've got a uracil as the starting base instead of a cytosine, a different tRNA is involved. So a little bit more than 20 transfer RNAs to bring in these amino acids. So when the process is occurring, after we have made that messenger RNA, there are multiple steps that need to happen in order for proteins to be produced. We start off with the initiation. So we can see our top sequence up here in pink, our messenger RNA. There's a little bit of it, a little bit downstream from our 5' end. That is the start codon, AUG. As our first tRNA recognizes this, there's going to be a bit of binding between the anticodon and the codon, bringing in this first amino acid. We talked about methionine as the first amino acid. And when it comes to prokaryotes, it's still methionine, but it's modified slightly from a regular methionine that we think about in eukaryotes. It's N-formal methionine. It's got an extra group, a methyl group attached to that. So it makes it slightly different, but it's ultimately the same idea. So the tRNA makes contact. The small ribosomal subunit recognizes and it makes contact. Once this has happened, the large ribosomal subunit sandwiches over that transfer RNA, and this is the initiation when it all gets linked up. And you can see our initial transfer RNA is in the middle position of this ribosome. This is where we're starting. As we start moving, as the ribosome starts moving along this messenger RNA, you see the arrow of direction. As we are moving through here, new transfer RNAs are coming into the ribosome in this first position. If they complementary pair to the messenger RNA, the growing amino acid or polypeptide chain, this string of amino acids, transfers over here, and we start growing this polypeptide chain. This is elongation. We're growing the protein. We keep progressing along the message until we hit one of those 3 stop codons. I mentioned before a release factor. It basically drives a wedge between the large and small subunit, causing the ribosome to disassociate, and we've got our completed protein sequence. At this point, we would call it a primary sequence. It's just a linear assortment of those amino acids. There's going to be all sorts of things happening to those amino acids once we've got that linear sequence. It will twist and fold, creating a secondary structure that folds back up on itself to create a three-dimensional structure. We call that tertiary structure. And with some proteins, that's the end of it. Other proteins, we think of multiple subunits, individual proteins that link up with others to create the actual functional protein. And in that case, we would call that a quaternary sequence. So sometimes it's tertiary for the final protein, and sometimes it's quaternary sequence, just depending on if there's multiple subunits participating in the function of that protein. So we mentioned before, there are definitely differences between this process and a prokaryote versus a eukaryote. Transcription and translation can happen simultaneously when we're thinking about a prokaryote. No nucleus, it's all accessible, so we start building the messenger RNA, and almost the instance that we've started to build the message, ribosomes can associate, and we can start building protein. And we have multiple ones going through this process, creating numerous products pretty much simultaneously. In a eukaryote, it's different because the nucleus harbors the DNA. This is where we make the messenger RNA. And I mentioned there's extra stuff going on within this DNA sequence in a eukaryote. And as we make that message, the initial bit is called pre-mRNA because it has all of the stuff, introns and exons. And the exons are what we're looking for. The introns are going to get chopped out of this. So we're not showing all of the details, but there's some processing, spliceosomes, bringing our pink bits that are what we want, bringing them together, and we get a little loop with the darker bit that gets cut out. Those nucleotides or those RNA nucleotides are going to get broken apart, degraded, and reused to build more stuff. But we don't want it coming down here. So we chop out the introns, and then we do a little more processing. Caps and tails get added, and I mentioned before, kind of like the zip code that's going to allow it to facilitate either with the rough ER or with free-floating ribosomes in the cytoplasm, dependent upon the fate of the protein. Once we've processed this, the messenger RNA leaves through some of the nuclear pores, and then it does those associations. And we can see a similar idea, like with our prokaryotes, where we can have multiple ribosomes and protein products being created, but not simultaneously, like we see in the prokaryote. A little bit different. So to compare these, the bacteria versus the eukaryotes, ribosomes are the workbenches. They are similar, but there are a few differences size-wise. Bacterial ribosomes smaller. You can see the two differing subunits. The S stands for Svedberg units, and it has to do with centrifugation. As we spin these down, they separate out and have differing weights. The intact ribosome, the two subunits bound together would have this specific size, but when we look at the individual portions, differing numbers, and they don't add up to the same number. As you see, 30 and 50s to give us 70, 40 and 60 to give us 80. So with that, the amino acids for our start codon, as I mentioned just a second ago, in bacteria, it's a formal methionine, and in eukaryotes, just methionine. Bacteria have a sequence in the messenger RNA called the Shine-Dalgarno sequence. It's present there, but absent in the eukaryotes. And this is just a little bit of sequence upstream from that start codon that helps to facilitate the linking up of those ribosomes. I mentioned before the interstate and the on-ramp and stuff like that. I really kind of feel like it's sort of a similar idea to that. This is allowing those ribosomes to make contact, um, so that when we get that initiation complex with the formal methionine, the ribosome can sandwich around it, and it's right in the middle. You back it up to this image, um, and we're not missing part of that. There's so a little bit over here would be the Shine-Dalgarno sequence that's helping these to link up. If we started right at the very beginning, AUG right here, our ribosome wouldn't have anything to really hang on to, so it needs a little extra space is kind of how I imagine. Uh, as I mentioned, no nucleus, no introns. It's all basically business. So transcription and translation can be simultaneous in the bacteria and definitely not happening in the eukaryotes. There's a lot of processing that needs to occur. So there are definitely potentials for problems when we think about uh, alterations in the DNA sequence. And I'm going to show you some images of things that potentially may lend to some of this here in just a second. We talked about the redundancy with that amino acid table. And this first bit right here really is an example of that. So this is what we call a silent mutation. We have had a change in the nucleic acid. In our DNA up here, we can see an adenine has been replaced with a thymine. We make our messenger RNA. The codons, the letters look different, but the amino acids they're actually coding for are the same. So you get a valine here, you've got a valine over there. Makes the same protein, same amino acids. So yes, there's been a mutation, but we don't see it. So silent, all right? And these, I should back that up, right at the top, it's a point mutation. So we're talking about a single base that gets altered, all right? We'll talk about some other potentials a little bit later. This next one is a missense. So we've got a different base put, or a nitrogenous base put in here that results in a change in that codon. So what should have been a proline is now a threonine. And I mentioned the properties of these amino acids with the way they twist and fold. Now the structure, the three-dimensional structure of this protein is not going to be the same and likely not functional. Not all mutations are bad. We oftentimes think of them as bad, and sometimes maybe something like this could confer some sort of an advantage and maybe a benefit to having had that occur. Truly depends. Uh, the nonsense mutations, these aren't a good situation. So in this case, we wind up, and it looks as if we're at the end of this sequence, but what should have been a tyrosine over here winds up being a stop codon that gets put in. And my assumption is we're only looking at 4 amino acids. There's probably about 85 more that could be linked up on here to make an initial small polypeptide. So we've only got the smallest bit and the stop codon shows up. So nonsense, you stop it where you shouldn't be, all right? That's a problem. These next ones are a little bit different, and it could be more than just a point mutation. We call these frame shift mutations insertions or deletions. Sometimes we describe it as an indel to use both words together. And this is inserting or deleting one or more bases. So up above, it's just a base that's getting substituted for something else. And here, we're adding or removing. And that ultimately sets the stage for everything after the point of that problem to be changed. Instead of just a single situation, you see all of these that are now different at this point. So cutting out or adding changes the reading frame. So reading these 3-letter words, and now we've shifted either adding a letter or removing a letter. And all of those words, those amino acids that will be following after that has happened, are jumbled up and no longer the same as we started with. Definitely problems. So I know there's a text box that goes through the littlest bit about this. Not always do we think of mutations as being bad news. I mentioned that just briefly with the last slide, that maybe it confers some sort of a benefit, which may make you wonder, well, why are we looking at HIV on this slide? What's this have to do with mutations? Interestingly, there are some people with a mutation. HIV wants to infect T cells, and it docks to specific T helper cells to receptors on those cells. And there are some individuals that have one of those proteins that has been mutated that no longer allows the docking of things like HIV. This is something that's occurred long before HIV was ever even on the radar. They speculate potentially something that maybe conferred an advantage to surviving the plague or potentially smallpox, maybe both, not allowing cells to be able to recognize those receptors and conferring some advantages. And interestingly, there are some uh, like Eurasian descent individuals that potentially harbor higher levels of these mutations, conferring them the ability to be resistant to this. I wouldn't go out and test that on yourself, but that is a potential that you may harbor a genetic mutation conferring potentially an advantage. I have learned some other stuff thinking about some of these same ideas that they speculate maybe those situations prevent something like this, but it may do the opposite and create other issues in the individual, like maybe enhanced likelihood of autoimmune diseases. And I think that is truly fascinating. We don't have all of those details figured out yet, but we're definitely learning more about that. And I think it's truly a fascinating topic. So, mutations can occur spontaneously. So, as we saw with like those point mutations, where you may see a base pair that doesn't get put down like it truly should. This could be happening in the process of DNA replication and something goes wrong. There are proofreading mechanisms that you would hope correct those mistakes, but sometimes it does not, resulting in those mutations. But there are other things that can speed that process up, if you will. So we've got a couple of different things we want to think about in this slide. 2-aminopurine nucleoside is an analog to adenine, and the 5-bromouracil is an analog to thymine, but they're not the same. And ultimately, what happens is this is going to cause some mutations to occur. The aminopurine nucleoside, that's the analog to adenine, A and T, right? This converts the ATs to GCs. It pairs with a cytosine, all right, instead of a thymine. And after a couple of rounds of replication, separating the DNA and creating the daughter sequence on there, semi-conservative process, after a few rounds of this, we have converted it, and now we have a base pair that's GC instead of AT. The opposite over here with the 5-bromouracil, it pairs with the guanines and converts an AT to a GC. So in both circumstances, we're getting guanines and cytosines where it should be adenines and thymines. And that in turn, point mutation causes, dependent upon where this is happening, differing amino acids. They may be silent or they may not, creating issues. Nitrous acid Our last bit down here at the bottom, it's a different type of agent that can cause damage, a mutagen causing damage at the DNA level. This nitrous acid modifies what's already there, uh, like the cytosines, and stimulates the production of uracils. That in turn will pair with an adenine instead of a guanine. So this converts GC pairs to TA pairs. Um, so kind of the opposite of what we're seeing here, but in both circumstances, not what you'd want to be having happening. There are other molecules that we could also think of as mutagenic agents that get into the DNA and cause issues within the sequence itself. Intercalating agents, acridine is a type of dye. We showed some of those fluorescent things. Acridine is a dye that will get in and do staining. Ethidium bromide we use for staining DNA as well. It also can get in and serve as an intercalating agent. So when they get into the DNA, it basically takes the space of a nitrogenous base pair. It removes them, if you will, insertions or deletions, as we talked about before. So that's what we've got here, a deletion or an insertion. And you wind up with differences. This is a frame shift mutation as we act as if this dye, this intercalating agent, is serving as a nucleotide, but it's not. So that has the possibility of causing some frame shift mutations, and everything downstream from that's a big problem. We can also think about radiation, X-rays and gamma rays. For our ionizing radiation cause damage to the DNA, breaking the covalent bonds in the backbone. We can see a single strand break, or we can even see a double strand break as we move over to the opposite end of this, causing damage to the DNA and potentially issues with the nucleotides as we're breaking through there. We're really just showing what's happening to the backbone. Ultraviolet light, the non-ionizing radiation, causes different damage. We're not breaking the backbone. What we're breaking are hydrogen bonds. The thymines and adenines that should be hydrogen bonding together, UV light breaks those hydrogen bonds, and it causes the two adjacent thymines to form a double bond, a covalent bond, between the two thymines. Once they're bonded up, they don't want to hydrogen bond, and you kind of get a little sort of speed bump in your DNA, a bleb, where these no longer make a nice smooth spiral because they're not binding like they should. Thymine dimers, definitely a problem, and there are potentials for cells to go through repair mechanisms. We'll see some of that in just a second. I think about my own skin as we think about UV light and the potential damage that it may harbor. And it can cause similar things within our own DNA, lending to these dimers. We have repair mechanisms to try to correct that, but it definitely can get overwhelmed. And that's where we start thinking about some potentials for skin cancer when these issues, these dimers, don't get corrected and lending to mistakes in the proteins that are trying to be generated, whatever it is it's trying to deal with. I skipped over that. So with our bacteria, there's two different mechanisms to repair these dimers. Up above, we're getting the damage. The UV light causes this dimer, and like I said, kind of like a little bleb, a little speed bump that shows up in the one sequence of DNA. So there is the potential for excision repair. This nucleotide excision repair recognizes this bleb, and we chop out the flanking region along with it, it gets removed, and DNA polymerase helicase is popping it out, and polymerase 1, our proofreader, uh, comes in and lays down new nucleotides. DNA ligase forms the covalent bonds in between here, gluing these back together, if you will. And now we're back to the repaired state. No more dimer going on. Nice and smooth. Another one is an enzyme photolyase, and this enzyme recognizes these dimers. It needs light, which I think is kind of interesting. Light was potentially the culprit for the damage, and now we're using it to stimulate the repair. This enzyme recognizes these dimers, and it's a similar sort of an idea, but we're not chopping out the nucleotides and replacing them. We're breaking those covalent bonds and allowing for hydrogen bonding to occur with their partners, as we see right here. So, bringing us back. Both of these mechanisms are in place in our bacteria. Some more active than others, just depends on the respective type of bacterium. But the capability for either gives them multiple ways that they can deal with this type of damage. This technique is something that we have done in the lab. We don't do it anymore, but it's something that we've done to basically try to visualize mutants, something that has been altered that gives us an opportunity to actually visualize it. So the replica plating uses a piece of velvet cloth. It is like a stamper that's going to transfer the bacteria from one plate to another, and then we will cultivate them up. One thing I'll point out, and we can kind of see it right here, there are some marks on the plate. Typically, I'd mark it so that my plate is right at like 12 o'clock and my mark's on there so I know this is exactly how my plate is. I've got them all set up exactly like that. And my tamper would also have a mark on it. So I have it all positioned right at high noon, and I stamp it onto the plate and I transfer it to another plate, and then I can make comparisons. As long as I've got something to signify all of these plates, orienting them properly, then I can look for what's happening. So what happens in here is we've got our media. In this case, we've got histidine that's incorporated into the media. All of our bacteria are happy and growing. We can see the colonies on here. Our velvet cloth, you can see the colonies are on it, and we're going to transfer it to these two different plates. One of them's got histidine, and the other one lacks it, all right? As we make these transfers, we incubate those plates up 37 degrees or whatever growth temperature they want to grow at. And then we look at them to see what's growing, or maybe better yet, what's not. So right here on this plate, there's a missing colony, but it's up above. All right. That tells us that this colony that we see on the plate that had histidine that isn't growing on the plate that lacks histidine has been mutated. All right. It now requires histidine to be incorporated into the media. And if you didn't give it histidine, it's not going to grow. So we've made a mutant. All right. These other cells that are growing in the lack of histidine are capable of producing histidine on their own. But this one needs us to supply it. All right. So it's been mutated. It keeps shifting on me. So this is really pretty interesting. And we did this experiment. We used the bacteria called Serratia. When Serratia grows at 25 degrees Celsius, it produces these pink colonies. We took the Serratia, we plated it out, we exposed it to ultraviolet light to stimulate a mutation, and then we did the replica plating. And it was the same sort of an idea. We used media that was like nutrient agar plates that have a lot of nutrients in there, and then a minimal media that's got a lot less nutrient. It's not just wasn't just one amino acid, but similar idea to look for stuff growing and not growing. But we made some observations in the process of doing this and recognized The serratia that turns pink at 25, those colonies, when they've been mutated, they lose the ability to produce those pigments, so their colonies are white. And you don't need to do any sort of replica plating to see that. It's just incubating them and seeing pink colonies versus white colonies. Pretty interesting. We also recognized that if we were to take those plates and leave them in the incubator for a little bit longer, some of those white colonies go back to pink colonies. And that is ultimately this repair mechanism where the damage has happened. We've lost the ability to produce the pink colony. And then as it gets repaired, the pink colony comes back. So not only were we witnessing mutations, where we're also giving it a little bit of time, witnessing the repair mechanisms that were happening as well. Pretty cool stuff. You'll still use some UV light. Now we use a couple of different cells and see some similar stuff. I don't think it's colony colors, but it's a similar idea. This one I think is really pretty interesting, and it made me stop and go back and be like, wait a minute, what's really happening here? The Ames test is a test that is designed to look for different chemical substances, different things that have the potential to be damaging to a cell, mutagenic, or even carcinogenic. Are these substances things that could be an issue? All right. And in order to test that, we use some bacteria. Easy to grow, easy to potentially mutate, if you will. And if some salmonella dies in the process, we're probably not too sad about it, right? So what's happening here is we've got our salmonella strain that requires histidine. It's already a mutant, if you will. It needs to have histidine incorporated into the media for it to grow. The next strain, also histidine needed, all right? And this first bit's a control, all right? So we're not adding the potential chemical agent, and we played it out, all right? And then we add the agent, and we played it out, all right? So there are potentials for some of these cells to mutate, and we call it a back mutation or a revertant. It goes back to the truly the wild type where it's capable of making histidine on its own. So you get a few cells that are able to do that. All right. And you kind of look at that, you count it up, and you're like, all right, in this, I've got a few that can do it. But as you compare it to the other plate, there's a boatload of them that have reverted back to histidine positive. So they're able to make histidine on their own. This agent caused damage and it caused mutations, and now they're able to grow. Little bit, whole lot. Problem with this. So the thing that made me stop was, what's up with the rat liver extract? Why are we adding rat liver extract to this? And the kicker is your liver does a lot of stuff, filtering and detoxifying, and the potential for it to maybe act on some of these agents and that might make it be the problem, was recognized. So adding a little bit of liver extract provides those enzymes within there that if it's something that needs to be processed through the liver, now we're ultimately giving it that. And that sets the stage for being able to visualize this. So the Ames test tests chemical agents for their potential to be damaging to DNA, whether it be mutagenic or carcinogenic or both. We would like to recognize that and know certain substances should use caution. So we want to shift over now and think a little bit about how bacteria can potentially pick up extra stuff. We've mentioned before that bacteria are asexual. They replicate by binary fission, copying their DNA, and they've got one cell's got the exact same stuff as another cell, but not always. So horizontal gene transfer is where we've got the addition of extra material. And there's three ways this can happen: transformation, transduction, and conjugation. Transformation, as we see here, we've got a plasmid getting pulled into this cell. That's okay, but I like to think of it as maybe like just a chunk of bacterial chromosome. This, a couple of different types of bacteria out in the environment. One bacterium dies, its cells all degraded, DNA is loose out there. Bacteria are very good at recycling. So if this bacterium comes up upon a cell who's released all this DNA, they may like to pull that in. There's a boatload of free nucleic acids here, and I can use those as building blocks for more of myself. But potentially, that chunk of DNA may be a full intact gene instead of just a few nucleotides long, and it could get incorporated right in to the chromosome of that bacterium. So transformation, we oftentimes call it the uptake of naked DNA. It could be a small little piece of it, or it might be a plasmid, just as we're showing right here. And a cell could pick up that plasmid, and now it's got something extra. Transduction uses a virus, so that virus harbors genetic material, and potentially some of that genetic material is actual bacterial chromosome, allowing it to get into the chromosome of a new cell, and now you've got extra information that you didn't have before. Lastly is the conjugation. So this uses a plasmid. The plasmid codes for the production of this pilus, a connector between this cell that's got it and the one that doesn't. We make a copy of the plasmid and pipe it over into the new cell. Now both of these cells have plasmids, and the plasmid bringing in some sort of extra information that can potentially stimulate, antibiotic resistance or tolerance to harsh chemicals or numerous other things. So all of these are mechanisms that bacterial cells, which are asexual, uh, have an opportunity to pick up new genetic information. So Vibrio cholerae is a pretty good example of some horizontal gene transfer. Cholera is bad news, and we're going to go through some details about that later on as we start thinking some GI tract stuff. But the issues with it are the toxin that this bacterium is able to produce, cholera toxin. But really, the deal is these bacteria produce the toxin because of horizontal gene transfer. And it's really some transduction. So a virus has inoculated them with this gene that gives them the ability to produce the toxin. This type of virus is a lysogenic virus, so it's one that incorporates into the chromosome of the bacterium, and then after, whatever sort of adverse condition may stimulate that part of the chromosome to be excised, we go back into the replication of the virus, and then they release out of the cell to go on and infect other cells. But while they're in the chromosome and within the bacteria, then we read those genes, we make those toxins, and the Vibrio cholerae that's got that virus within it is the one that causes the super ill effects as a result of it, because it's harboring that extra suite of genes that say, make this person super sick. Pretty crazy. So this is more conjugation. So we've got an F plus and an F minus cell. The F plus meaning it's got the plasmid, and the minus it lacks it. So the plasmid here, this F plasmid, F stands for fertility. It can harbor, just as I've described before, all sorts of different capabilities to survive, withstanding antibiotics or other things, but it also has the gene for conjugation, for the formation of this pilus. So the cell that's got it in close proximity to another cell, and they don't even have to be the same species, just close enough, all right? A pilus forms. We ship over a copied piece of that plasmid into the minus cell, and what was once a plus and a minus becomes two plus cells, as we see here, both of them harboring that plasmid. There are potentials that we will see shortly where maybe we get a little bit more and we bring some bacterial chromosome along with the plasmid, then we're a little bit different, all right? So this typical, uh, but there are other issues, all right? So the HFR stands for high frequency of recombination, all right? So here we've got our cell, we're, we've conjugated, we've done all of this, but the plasmid inserted into the chromosome instead of hanging out in the cytoplasm by itself, all right? So now this cell is an HFR, or a high frequency of recombination. We also call these cells promiscuous because they would like to conjugate all of the time. Uh, that's kind of the idea. If this cell wants to replicate every 20 minutes, it would also like to conjugate every 20 minutes, all right? So here we can see it, and this we've got this lack for lactose to be able to utilize that or not. That's this little gene that was on the plasmid, I'm assuming over here, all right? Uh, so it gets inserted along with that plasmid, and now we've got this cell that harbors it, all right? And over here, our plasmid is bringing with it that gene. This bit was coming from the blue. Now it's got that lac bit right within it, all right? Giving potentially somebody completely different the ability to utilize lactose when it never had that potential in the first place because it's bringing that suite of genes with it, giving them truly some variation that they never had before. So this clock over here is basically showing the sequence of the plasmid itself and specific genes within there, things that we might be able to manipulate or see it using something or not, right? And knowing this timing, which is like 100 minutes as we move around this clock, um, you can see what's happening with these cells, these HFR cells, and the transfer as it's moving over, right? Um, Piping into those cells. So, like I said, about 100 minutes for the entire genome to be piped over um, into a new cell. Knowing a little bit about the plasmids, a little sidebar from these HFRs, knowing where some of these genes are, you can make specific cuts in the plasmid, inserting genes that you may want to be incorporating in this similar process, just like this, creating cells that now harbor something that you're doing research with, trying to figure out details about what's happening. Another mechanism that little bit different than the three that we've described is some jumping genes, transposons. This enzyme transposase, sorry, this this enzyme transposase can chop out this gene uh, and move it to some other region on that chromosome. When we're talking a prokaryote, when we're talking eukaryotes, it could go to a completely different chromosome altogether, creating bigger problems. Not that this isn't a big problem. So you can wind up with genes moved to completely different regions that can also lend to things like insertions, where we've now got extra sequence in right in the middle of a gene that now we've got stuff that we don't need. And as you see over here, disrupted because our gene B is now twice as big as it should have been, right? With all of this extra information. But they do have the potential to incorporate information that may, in some circumstances, hold some sort of a benefit. So in prokaryotes with a single chromosome, most all of the genes needed for whatever process are basically right beside one another on the DNA, and we call it an operon, where it's got all of the stuff needed, right? And you don't see this stuff in eukaryotes. You don't see these operons with all of the genes together. Sometimes they may be on completely different chromosomes, right? So promoter, operator, and then the business of what we want here in green. This is what we want to be making. So all of this is our DNA. We've got a little bit of regulatory stuff here that we'll hit on in a second that may stimulate whether or not we're going to be doing this or not. Maybe we want to make the mRNA, maybe we don't. All right. And the regulatory genes can dictate whether or not this is going to be accessible or not. Promoter, allowing a little bit of sequence for where our RNA polymerase can come in and recognize to lay down that sequence. To start building our message, all right, for the RNA polymerase to link up on there and start building it. The operator will see more details with this in a second, with some regulation with this that could be turning on or turning off the production of these, the transcription and the production of these proteins. So we build this message, we make this entire copy, and from that bit, these 4 different genes, we've got, in this case, 4 different protein products that we are producing. And we're making them all at once. We need all of these things to utilize lactose or tryptophan, both of which we're going to talk about as we move through the last little bit of this. So first off, tryptophan. Bacterial cells have the potential to be making their own tryptophan, right? They can do it. They've got genes that allow them to make it if they need, all right? So here's our promoter, the polymerase linking up, our operator, and everything's all fine and good. And you see down below the repressor is not linked up, all right? The repressor is like the off switch. So if tryptophan isn't present in the media, this bacterial cell's like, well, I need to make that. All right. And the repressor falls off and synthesis proceeds. We make the messenger RNA, we build the respective proteins, and now we've made ourselves some tryptophan. If we're growing in a medium that's rich with tryptophan, that cell doesn't need to waste its time making its own tryptophan because we're giving it to it. So in the presence of tryptophan, the repressor binds, and it binds because tryptophan links up to it, causing it to stick to that operator region, basically shutting down the activity of polymerase. You can't move past my operator bit, my off switch, right? Prevents that from happening. So this one depends on the circumstances, all right? If there isn't any tryptophan, the switch is off, is basically the repressor is off, the switch is turned on to make it. If tryptophan is present, the repressor binds, the switch is turned to off, and we don't make any of it. Lactose is a little bit different, all right? It's a similar idea with these repressors, but this one's different. We're not making lactose. We're making the enzymes needed to break down lactose. So if there isn't any lactose in the media, a repressor links up to the operator and says, why waste your time? We don't even have milk up in here. So we don't do any sort of transcription and no translation. We're not going to make those enzymes. But if lactose is present, the lactose combined to the repressor, same sort of an idea, causing it to drop off, and now we're turned on to build those enzymes needed to break down lactose, all right? So Depends on the situation. Uh, Tryptophan, we can build on our own. If we've got a lot of it, we're not going to make it. Lactose, we'd love that sugar, but we're not going to waste our time building the enzymes unless the sugar's present. Technically, there's a little bit more to it than that, and I'll show you that in just a second. So in this example, we've got some E. coli. I've already mentioned E. coli loves lactose, right? But glucose is a monosaccharide, simple sugar, right? Lactose is a disaccharide. So it's easier to use glucose, and it will have preference to use the glucose first if we're growing in the presence of both. So E. coli, time-lapse photography over here, we've got 10 hours or so. Um, we start off a little bit of a lag phase. We start using that glucose, and we can see E. coli growing really pretty well. In first few hours, it pretty much uses up that glucose, and we see another kind of lag phase. This little bit that we see here is basically this stuff that's starting. We're needing to build these enzymes to use the lactose. So it takes a little bit of time because lactose is there. We've linked up to it. Now we make those enzymes and then we start using the lactose uh, and continue to grow for the rest of the, half a day in this tube. It's not maybe quite as dramatic of a growth curve. It's a little bit less, not quite as steep, but still pretty good growth, right? Um, so there's a few things that stimulate it, and as it's just growing right here, with both of them in the mix, we're not making the enzymes needed to use the lactose until we're about to the top of this right here, and then we're going to start dealing with it because there's ample glucose. So why waste your time on the lactose when I'm still bathed in sugar, right? So more than just lactose being something to stimulate it, ATP gets involved and it's going to help this process. So here's our ATP molecule. When these levels start to decline as glucose starts to get, used up, some of that ATP gets converted to cyclic AMP, and two phosphates, pyrophosphate, get cleaved off of that. Adenyl cyclase removes those phosphates, and then you've got this cyclic AMP. It's another switch, all right? And those cells are sensitive to cyclic AMP, stimulating them to recognize depletions in energy sources. Glucose, right, has gone down. What are we going to do about it? All right. So, a couple of things going on here. This first bit, one bit, and the next one. All right. So, if we've got our cyclic AMP, all right, uh, we'll start up here. All right. Uh, in the absence of cyclic AMP, CAP does not bind to the promoter, and transcription occurs at a low rate. We're doing it, but not like we could be. But if we've got cyclic AMP, then the cap binds, and then we truly enhance this activity. And you see a big boulder arrow. That's when we really start making multiple messages very rapidly. So 'cause we're like, we got to use some lactose. All right. That's the idea with this. All right. Um, so we can use that. And there's other potentials besides just lactose that may be functioning like this. All right. As we move over to frame B, for this black operon really to be expressed, we need the cyclic AMP and the cap, um, but we also need the repressor to get removed. So lactose is what's going to link up to this and cause it to fall off, and then we can move. And that's what's happened up here. All right. Um, so as long as there's lactose and we've got both of these up here linking up to it, we're going to go through here very rapidly. But down below, even in the presence of this, if there isn't any lactose, it doesn't matter. We've ran out of energy. We've depleted the glucose reserves, but there isn't lactose. I'm not going to make this just because I ran out of glucose. If lactose isn't there, we're still repressing it. All right. Um, that's the idea with it. So this one I've got a little bit of an issue with because I feel like it isn't quite accurate. And I went back to the text and read through there to see if maybe I was mistaken, but I think I was not. I think it's a little bit wrong. So tryptophan plentiful. Remember, the cell can make it on its own, uh, but if it's plentiful, why are we going to waste our time? All right. So that's what we're thinking about here. All right. So we go through building this messenger RNA. Our polymerases uh, going past here, our ribosomes. We're building proteins. We're not showing the proteins coming out, but that's the idea. All right. So we're up at the top, we've got high levels of tryptophan and low levels of tryptophan, and this would be in the media. All right. So if we've got high levels of tryptophan, we don't want to be going through this whole process. And this is where I've got part of the issue. You see a complete polypeptide and ribosome preceding. I think this needs to be down here, right? It's where it's not. Well, the complete polypeptide and ribosome preceding needs to be down here, and the incomplete and ribosome stalls needs to be up above. We can't make it here. We've stopped because of this terminator stem loop. The anti-terminator stem loop, which is just more confusion, allows for that to proceed through. And you see transcription proceeding, translation would be proceeding, here transcription's terminating, and translation should be terminating as well. So these two things should be flip-flopped, all right? If there's low level of tryptophan, all right, in that media, we would like to make this sequence. Confusion? Hopefully I worked you through that stuff. Up above, high level of tryptophan, we shouldn't be building this complete sequence. We should shut it off. As soon as we hit this, we should be done. Low level, we want to keep going. So it's not going to stall, it's going to proceed. This will stall. Ribose switches are kind of the same idea as what we were looking at with those little hairpin loops right there, allowing things to proceed or not. So up above, we can see in the on situation, we're allowed to move through there, polymerase making the message, ribosome going through and building those peptides. And when the ribose switch is basically shutting it down, the loops go together, as you see here, and there's molecules that may facilitate this acting like the switch, preventing the translation from occurring on this messenger RNA. That's the idea. And that's what we're looking at here, messenger and DNA down below. We're just not showing the DNA in these sequences. So in eukaryotes, there's an enhancer, it's a DNA sequence that helps to promote transcription, and it's a short bit of DNA. Activators bound to this help to mediate this process of transcription, lending to it being able, and that's what we're seeing up here with these activators linking up in this enhancer region that allows it to move through this process. Transcribing, make a messenger RNA, these respective genes. And if our activators aren't coming up and linking up to this, we're not going to be producing those respective products. A different way that we think of as far as control mechanisms go. Okay, that is the end of 11. I'm going to stop it there. I will see you all back in person for real on Friday. Farewell and have a good day.